we've gone and sequenced all of the genes in the cell, but in reality, in most experiments, only 2,000 to 3,000 of those are actually going to be changing in response to whatever biological condition you're looking at. And the other 10,000 are just going to be subject to technical noise. So if you don't, if you use the entire expression matrix, you have to be very careful that whatever method you're using isn't just picking up the noise in all those 10,000 genes, rather than the biological variants that you're interested in the sort of 3,000 gene range. Because many methods can be sensitive to the large amount of technical noise, it's often easier to just select the 3,000 or so genes that you think are responding to biological differences ahead of time and only consider those for your downstream analysis, and then throw away all the other 10,000 that are probably just technical noise. So that's what we're trying to do when we're identifying important genes. So the first method we're going to look at is M3 drop, which is based on modeling the dropouts within single-cell RNA-seq data. And we're going to use this Usoskin data set, which is looking at different types of neurons to do it. So you can load the data and see that we have 4 different types of neurons in this data set that they've annotated using marker genes. And then we're going to fit the M3 drop model, which uses the Michaelis-Menten equation, which is based on the fact that the limiting step in the single-cell RNA-seq process is the reverse transcription. Reverse transcription is much less efficient than PCR amplification, and it's because it's the very first step. If it fails at the reverse transcription step, you're never going to get any reads from it at the end. So it's the main source of the zeros in most single-cell RNA-seq data sets. So this is the Michaelis-Menten equation, where the probability of being a dropout is related to this constant that we're going to fit, and S is the expression level of the gene. And there are a couple of other models that have been proposed in different papers. One is this logistic model, and the other one is this double exponential model. Or again, S is the expression level of your gene, and lambda is a parameter, and there are 2 parameters in the logistic regression. So using M3 drop, we can fit all of the different models to this data set. And you can see that in red, the double exponential model doesn't fit very well at all, whereas the logistic regression and the Michaelis-Menten are almost the same. Because of the simplicity of the model, we're going to be using the Michaelis-Menten function. So now that we've fit this model, you can find that, so before we were looking on the log scale, here we're looking on a linear scale. Again, the black line is the Michaelis-Menten fit. So here if we've got a gene that is very highly expressed in one subset of cells, it's going to have very high expression and very few dropouts. But if it's lowly expressed in a different population of cells within our data set, that's going to lead to very high dropouts and low expression. And when we average across those, we're going to end up somewhere shifted to the right of the curve because of the shape of this curve. Hence, in order to identify differentially expressed genes, we can just identify outliers shifted to the right of the curve. So you can use this function in M3 drop, which implements a test, which basically calculates a k-value for each individual gene based on its expression level and its dropout rate, and then compares that to the fitted k across all of the genes, again, accounting for all the sources of error and all of these different things, and then tests for whether it's significantly shifted to the right of the curve. So you can see we find a whole bunch of genes that are shifted to the right, which are likely to be differentially expressed. So in order to check this, we've got a whole bunch of neuronal markers because this is a neuronal data set. And we can see how many of those have been detected as differentially expressed using M3 drop. And you see that almost all of them have been. So there's 2 that haven't been, but the rest have all been detected. Another way of validating that this differential expression has worked well, if you don't have known markers for whatever system you're looking at, is to just plot a heat map of the gene expression level. So there's the function that we've provided to do this. And when you do that, here we've got the known cell types colored at the top, and the expression level of all of the genes in the matrix. And you can see that you get these very nice blocks of gene expression where the gene is highly expressed in one particular subtype, subpopulation, but lowly expressed in the other subpopulations. And you see that just using hierarchical clustering that was talked about in the previous section, you can recapitulate the different cell types very easily in this data set. So this is our method that's based on looking for differentially expressed genes using the dropouts. But there are other ways of doing this. The other main method is published by Brunecki et al., which usually uses spikins to model the technical noise and then look for genes that are significantly above that as having significant biological variation that would be of interest. However, because it's reliant on spikins, and we've now found that spikins actually don't model technical noise of endogenous genes very well, I've modified this method in M3 drop below to use all of the genes to fit the technical noise model, and then look for genes, those genes which are significantly higher than the rest of the genes. Yes. Basics also depends on spikins. So yes, it uses the spikins as the prior in order to distinguish biological and technical. So you also need quite good quality spikins to use basics, but it's also much, much slower than the Brunecki method. And when I've compared the 2, you get almost the same thing. So again, here we fit the null model, here using all of the genes rather than the spikins, because there weren't spikins in this data set. And then in pink are all those genes that were just called as having significant biological variation. And you see it, we find 1,500 genes using this method. So as an exercise, you should plot the heat map of these differentially the highly variable genes, as well as check the marker genes that are identified using this method to compare against M3 drop. And then for you, we've compared this also with RNA-seq, which we provided because it takes quite a long time to run. So we have the RNA-seq run on the known different subpopulations in this data set, the highly variable genes method, and the genes picked up by M3 drop. You can see that almost all of the genes identified by M3 drop are also identified by one of the other methods, whereas the other methods have a lot of genes that are very unique to them and less overlap with each other. Yeah, M3 drop is more conservative than the other methods. And if you look at the heat map, so you should produce the heat map for the highly variable genes, and you should be able to see that a lot of those genes do not look like they're actually differentially expressed between the subpopulations. it's also because variance is actually a very noisy measure for single-cell data, particularly for, so once you get below 500 counts per million for gene expression level, then variance becomes very unstable, and you get a lot of sampling noise. So you get a huge number of false positives when looking for highly variable genes for anything below sort of 500 counts per million, whereas dropouts is a lot more stable for lowly expressed genes. That was, I'll give you a few minutes to plot the heat map to compare the highly variable genes and the known markers.